Welcome back. In this explainer, we're diving deep into the otolith organs, our body's very own gravity sensors. We're going to tackle the central paradox they present. How does our brain know the difference between tilting our head and moving forward, when the physical signal it gets is almost exactly the same?
Okay, let's try a little thought experiment. Picture this. You're in a car and it's accelerating forward. You feel that push back into your seat, right? Now imagine you're just standing still and you tilt your head back. Believe it or not, to your inner ear, the physical force from those two totally different movements feels almost identical. This is what we call the gravity translation ambiguity. It's a huge puzzle. So, how on earth does the brain figure out what's really happening? Well, it all starts with the sensors themselves.
And those sensors are the utricle and the saccule. These two little otolith organs are our primary detectors for what's called gravito-inertial acceleration. That's just a fancy way of saying the combined force of gravity plus any linear motion your head makes. What's really cool is that their physical orientation completely dictates what they do. The utricle, for instance, lies flat. It's horizontal. This makes it perfect for sensing any forward and backward motion and side-to-side head tilts, what we call roll. The saccule, on the other hand, is oriented vertically, kind of sitting up in the sagittal plane. This makes it the go-to sensor for up and down movements and, of course, the constant pull of gravity. So how do they actually detect this force? Well, each organ has a little patch called a macula. This is covered by a gelatinous layer that's loaded with these dense little crystals called otoconia. Now, because these crystals are heavy, they've got inertia. So when your head moves, they tend to lag behind, creating the shear force that actually bends the tiny hair cell bundles underneath. And that bending, that's the mechanical trigger for a neural signal.
Well, just detecting motion is one thing. Detecting the direction of that motion with incredible precision is a whole other challenge. Nature's solution for this is just elegant. It's something called the line of polarity reversal, or LPR. Think of it as a functional dividing line that runs right down the middle of each macula. On either side of this line, the hair cells are oriented in completely opposite directions. It's fascinating. In the utricle, they're all polarized towards this line, which we call the striola, but in the saccule, they're polarized away from it. So what does that do? It means that any single movement is going to excite the hair cells on one side and at the exact same time inhibit the cells on the other. It's a perfect push-pull system. This is an amazing biological trick that dramatically enhances directional sensitivity and basically doubles the organ's dynamic range.
This brings us to another really fascinating part of how the otoliths work. These organs have to sense the constant, super low frequency pull of gravity, day in and day out. But they also have to respond to very high frequency vibrations, like sound that's conducted through your skull. How can one single mechanical system possibly do both of those things?
The answer is all about frequency-dependent mechanics. See, at low frequencies, like when you do a slow head tilt, those heavy otoconia have time to shift and drag across the hair cells. We call this accelerometer mode. But at high frequencies, think a quick tap on the head or a burst of sound. Those heavy otoconia stay pretty much still because of their inertia. Instead, the skull and the macula vibrate beneath them. This is what we call seismometer mode.
And this isn't just some cool biological fact, it's the entire foundation of a key clinical test. This seismometer mode is the exact principle we use for vestibular-evoked myogenic potential testing, or VEMP testing, where we use sound or vibration to specifically activate this high-frequency pathway. And this lets us isolate and test very specific parts of the otolith system. For instance, a cervical VEMP, or C-VEMP, tests the saccule and its connection through the inferior vestibular nerve, and an ocular VEMP, or O-VEMP, tests the utricle and its pathway through the superior vestibular nerve. It's an incredibly powerful diagnostic tool that comes directly from this amazing dual-mode property. Okay, so let's boil this all down. What are the key clinical takeaways here? Honestly, understanding these mechanics is absolutely essential if you're going to interpret tests like VEMP, the subjective visual vertical, or the ocular tilt reaction. First, and this is huge, the otoliths themselves cannot tell gravity and linear acceleration apart. That job falls to the brain, which has to integrate signals from the canals and our other senses to figure it out. Second, that line of polarity reversal creates an internal push-pull system, which is just a masterpiece of biological engineering for supercharging directional coding. And third, remember that frequency is everything. The stimulus frequency determines the organ's mechanical mode, and that's the entire physical basis for why we can use VEMP testing to assess otolith function.
So, we've covered how the force gets detected. But how does the physical bending of a hair bundle get turned into a neural signal at almost unimaginable speeds? Join me in video 5, Hair Bundles and MET, Tip Links and Microsecond Gating. We're going to find out how this process happens in under 10 microseconds, and why this ultra-fast transduction is the absolute non-negotiable requirement for a vestibulo-ocular reflex that beats our visual processing by more than 100 milliseconds. Thank you for listening to this series. To explore more similar topics regarding dizziness, vertigo, and imbalance, and to get clinician and patient resource materials, visit AustralianDizzinessClinics.com. Please subscribe and leave a rating.